A Stockholm University geneticist stands in front of a lab full of thousand-year-old teeth and delivers the sentence that undoes a national myth. The Swede doesn't exist genetically. This isn't a claim from a fringe blogger or an internet provocateur. It comes from Professor Anders Götterström, speaking after his own team's DNA results from the town of Sigtuna came back. And the scale behind that sentence is staggering. 442 Viking-era skeletons sequenced across 6 countries in a single nature study alone, followed by 297 more in a second study spanning 2,000 years of history. These aren't small sample sizes gathered from one grave or one region. This is a continental-scale genetic accounting of a population that a nation built its identity around. The results didn't just complicate the story of the Vikings. They contradicted the exact, pure bloodline image that has been used, for over a century, to justify some genuinely ugly things. That contradiction is the heart of this episode, and it raises a question that goes far beyond archaeology or ancient DNA. If half a Viking-age Swedish town turns out to have been foreign-born, what does that mean for the word Swedish at all? What does it mean for any nation that has built its sense of self on the idea of an unbroken bloodline stretching back to the age of longships and raids? So what happens when a nation's ancient DNA turns out to tell a completely different story than the one it grew up believing? That's the question this episode is going to answer, piece by piece, genome by genome. And the answer doesn't start in a museum, with a placard and a glass case. It doesn't start in a university lecture hall either. The answer starts not in a museum, but in a boat, at the bottom of the Baltic Sea. Before we get to the part with the swords and the scandal, it's worth sticking around for this one. If ancient DNA quietly rewriting national mythology sounds like your kind of story, there's a lot more of it coming, and hitting subscribe means you won't miss where this trail leads next. Because it leads somewhere strange. A cold fact, dropped straight in. For well over a century, the popular image of a Viking has been remarkably consistent. Blonde hair, blue eyes, horned helmet myths aside, but still an ethnically uniform band of Norse raiders, united by blood and by birthright. This is the picture on movie posters, on tourist merchandise, on the covers of history books sold in airport shops. A single people, a single bloodline, a single story. That picture didn't come from the Viking Age itself. It came from somewhere much more recent. National Geographic's 2020 coverage of the Viking genome studies traced the pure Viking bloodline myth directly to nationalist movements of the late 19th century, not to the 9th or 10th centuries when actual Norse seafarers were sailing, trading, and raiding across Europe. The myth is a modern invention, retrofitted onto an ancient people who never asked for it. Even before DNA sequencing entered the picture, archaeologists and historians had reasons to suspect the tidy, uniform version of the Viking story was wrong. The sagas themselves describe seafarers with complex, mixed lineages, families and crews that didn't fit neatly into one ethnic box. The written record was already hinting at something messier and more interesting than the myth allowed. And the myth didn't stay in the past. The same pure bloodline idea is still invoked today, actively, by white supremacist groups claiming a kind of Viking superiority, using Norse imagery and ancestry claims to justify modern ideologies of racial purity. So what were the DNA studies actually built to test? Not romance, not legend. Ancestry, movement, and mixture, gene by gene, grave by grave, across centuries of Northern European history. And that sets up the irony driving this entire episode. The DNA didn't confirm the myth. It dismantled it. It dismantled it using genomes recovered from the Vikings' own graves, pulled from their own teeth and bones, unable to lie the way legend and nationalism had. The mainland believed the myth. The genome didn't. A hook-style line to open this chapter. One paper changed how scientists talk about the Viking world, and its name is Margarian et al., 2020, published in Nature 585, under the title Population Genomics of the Viking World, led by geneticist Eski Willerslev. The scale of the project alone sets it apart. 442 ancient human genomes, sequenced from remains recovered across Greenland, Poland, Russia, Scandinavia, the United Kingdom, and Ukraine. This wasn't a study confined to one country or one archaeological site. It was an attempt to map an entire diasporic world through DNA. The time frame centers on 750 to 1050 CE, the classic Viking Age, though the researchers extended their sampling back into the Bronze Age to establish context for what came before. That long baseline let them see change over time, not just a single snapshot. The headline result reshaped the entire picture of Viking migration. Danish ancestry flowed into England. Swedish ancestry flowed into the Baltic region. Norwegian ancestry flowed into Ireland, Iceland, and Greenland. Three distinct, geographically separate migration streams, not one homogeneous Viking wave sweeping uniformly across Europe. The Vikings, genetically speaking, were never one people moving in one direction. One detail from the study directly undercuts the assumption that Viking equals Scandinavian. Two skeletons buried in Orkney, Scotland, in full Viking sword, Viking-style graves, complete with the weapons and burial customs associated with Norse warriors, turned out to be genetically Irish and Scottish. People who lived, fought, and were buried as Vikings without a drop of Scandinavian ancestry. The team also uncovered something more intimate: genetic evidence that some Viking raiding parties literally included close family members, not mercenary strangers thrown together by circumstance, but brothers and kin, sailing, raiding, and dying together. One of those family groups turns out to be the best documented brotherhood in the entire dataset, and it's buried in Estonia. A cold scene to open. 2008, on the Estonian island of Saaremaa, workers laying an electric cable strike something hard beneath the soil. It isn't rock, it's human bone, and Viking-age weaponry, buried near the village of Salme. What followed, in excavations running from 2008 to 2012, was the uncovering of two ship burials. Salme held the remains of 7 men. Salme II held 34. Both were dated to roughly 750 CE, a date that places them at the potential very start of the Viking Age itself. Some 50 years before the raid on Lindisfarne, in 793, that traditionally marks the era's beginning. The larger of the two vessels is the earliest known sailing ship to have crossed the Baltic Sea, a find of enormous significance on its own, well before any human remains were even considered. Every individual recovered from the two burials was aged between 18 and 50. Skeletal trauma patterns across the group point to violent deaths, consistent with a raiding party that went badly, catastrophically wrong. Strontium isotope analysis of the men's teeth pointed toward a specific home region, Swedish upland, with researchers noting a real possibility the men came from Valskerda itself, or from a nearby center of power in that region. Then came the twist. Some of the buried men's childhood isotope signatures didn't match upland at all. Instead, they traced back to the island of Gotland. The crew wasn't drawn entirely from one place before they became a crew together. They were already a mixture of origins long before they ever boarded that ship. The isotopes named a region, the DNA named a family. Ancient DNA analysis, built on the Margarian et al. 2020 dataset, revealed something remarkable about the men stacked together in the Salme II burial. 4 of them were full brothers. A 5th individual, buried close by, was genetically close enough to be an uncle. This wasn't a crew of strangers assembled for a voyage. This was, at its core, a family. Recovering that information from thousand-year-old bone wasn't simple. Geneticist Marie Allen of Uppsala University has spoken to the technical challenge involved. Ancient DNA degrades into tiny, fragmented pieces over centuries, and only the best preserved teeth and temporal bones yielded sequences usable enough to work with. Every result from Salme came from a narrow window of surviving material, painstakingly recovered. The Y chromosome haplogroups pulled from the Salme dead carry their own names in the genetic record: R1A1A1B, N1A1A1A1A1A1A1A, and I1M253. On the maternal side, the mitochondrial haplogroups recovered were T2B5A, V, J2A1A1A2, H10E, K1C1H, W6A, and U3B1B. That's a wide spread of lineages, wide enough to show that beyond the brothers themselves, this crew wasn't built from one single tight bloodline. And yet the rest of the Salme dead weren't strangers either. DNA confirmed that the remaining men came from the same district as the brothers. This was a raiding party built on real, local, overlapping kinship networks. Neighbors, cousins, in-laws, people bound together by geography and family long before they ever set foot on that ship. Not conscripted strangers pulled together by force or circumstance, but a genuine community that went to sea together, and in Salme's case, died together. That district has a name: Upland, the region strontium isotopes had already pointed to before the DNA confirmed the family ties within it. It's a place, a home base, a source of kinship and crew alike in the earliest years of the Viking Age. And that district, Upland, shows up again 2 centuries later, in a very different kind of grave, one that has nothing to do with brothers, kinship networks, or a ship lost in the Baltic, and everything to do with an assumption that stood unchallenged for well over 100 years. A cold fact to open: Birka, Sweden, holds over 3,000 known Viking-age graves. Roughly a third of them have been excavated, making it one of the largest concentrations of burials anywhere in the Viking world, a site archaeologists have returned to again and again for well over a century. One grave among those thousands became something close to a textbook illustration. Grave BJ 581 was excavated in 1878, and for well over a century afterward, it was treated as the definitive example of a high-status male warrior burial, the image reproduced in books, museum displays, and academic papers alike. The grave goods explain why. Inside was a sword, an axe, a spear, armor-piercing arrows, a battle knife, 2 shields, and 2 horses, a mare and a stallion. This was the complete kit of a professional warrior, assembled with a completeness rarely seen in a single burial. Alongside the weapons sat a full set of gaming pieces, the kind used for tactical board games of the period. Researchers read this detail as evidence the person buried there understood tactics and strategy, a signal of rank, marking them out as a high-ranking officer rather than a common foot soldier. For more than 130 years, one crucial assumption about this grave was never actually tested. The sex of the occupant was inferred entirely from the weapons surrounding them. Swords and spears meant a man. No one checked the bones themselves to confirm it, because the grave goods seemed to say everything that needed saying. Then, in 2014, an osteological reanalysis of the skeleton raised the first real doubt. Examined directly, the bones themselves looked biologically female, a conclusion that sat awkwardly against more than a century of assumption built entirely on iron and steel. The bones had been suggesting this for years. The genome would settle it. In 2017, a team led by Hedenstierna Johnson published a paper whose title alone was enough to unsettle a century of assumption. A female Viking warrior confirmed by Genomics, released in the American Journal of Physical Anthropology. The team didn't rely on a single sample. They tested both a tooth root and an upper arm bone from the Birka skeleton, and both returned the same result: 2 X chromosomes and no Y chromosome present anywhere in the genetic material. There was no ambiguity left in the data. The mitochondrial haplogroup recovered was T2b, a lineage common across England, Iceland, and Scandinavia, situating the individual within a broad and familiar swath of the Viking-age genetic landscape. Strontium isotope analysis of 3 molars added a mobility profile to the genetic picture. Sometime between roughly ages 4 and 9, this individual moved from an unidentified homeland to Birka, with the isotope signature pointing most likely to southern Sweden or Denmark as her place of origin. She wasn't born at Birka, she arrived there as a child. The researchers stated their conclusion directly, without hedging. The individual in grave BJ 581 is the first confirmed female high-ranking Viking warrior. A sword, an axe, a spear, 2 horses, and a set of tactical gaming pieces, all belonging to a woman. The finding triggered real academic pushback. In 2019, a reassessment published in Antiquity by Price and colleagues defended the original interpretation in detail, responding directly to archaeologists who argued that weapons found in a grave don't automatically prove the person buried there actually fought with them. The debate wasn't about the genetics, which were clear. It was about what the genetics meant. Birka answered one question about who could be buried as a warrior. Sigtuna, a few decades later, would answer a much bigger one about who counted as Swedish at all. A cold fact to open: Sigtuna stands as one of Sweden's earliest formally planned urban centers, a Christian-era town occupied from the 10th through the 12th century, built during a period when Scandinavia itself was transforming. In 2018, Krasinska and colleagues published their findings in Current Biology, drawing on genome-wide data from 23 individuals and strontium isotope data from 38 more, all recovered from Sigtuna's cemeteries. The result reshaped what founding-era Swedish town actually meant. Genetic diversity among Sigtuna's residents exceeded that found in Neolithic and Bronze Age European populations, and in the study's own comparisons, it even exceeded the genetic diversity measured in modern East Asian populations. A single medieval Swedish town, more genetically diverse than entire eras and regions. That level of diversity put Sigtuna on par with Roman soldiers stationed in Britain, a population already famous among archaeologists for its cosmopolitan mixing of peoples drawn from across an empire. Roughly half of the tested townspeople had grown up locally, around the shores of Lake Mälaren. The other half came from remarkably far afield: Ukraine, Lithuania, northern Germany, the British Isles, and Central Europe, alongside southern Sweden, Norway, and Denmark. Sigtuna wasn't a closed community. It was a crossroads. Two of the Y chromosome haplogroups recovered from Sigtuna's dead, I2A and N1A, don't appear in modern Sweden or Norway at all. Instead, they're linked to Eastern and Central European populations, and to Finno-Ugric groups, lineages with no continuous presence in the region today. Half the people buried in one of Sweden's founding towns weren't born Swedish by any modern definition, and one scientist said so, on the record. Anders Götterström, professor of molecular archaeology at Stockholm University, gave the study's conclusion to the TT Newswire in plain, unadorned language. It was a sort of Viking-age Scandinavian Shanghai or London. His follow-up line is the one that gives this episode its title thread. "The Swede doesn't exist genetically," he continued, laying out the implication without softening it. "We've pieced ourselves together from parts taken from the whole world, and the more this gets studied genetically, the more it becomes clear that people have been moving around the place the whole time." This wasn't a marginal press release buried in an academic newsletter. It ran through Swedish national media, reporting on a peer-reviewed paper published in Current Biology, carrying the full weight of institutional science behind a statement that directly challenged a national self-image. Set this beside the earlier Margarian finding from Orkney, the two Scottish and Irish skeletons buried in full Viking warrior style. The pattern is identical, only the country has changed. Viking identity and Scandinavian ancestry keep turning out to be two entirely separate things, tangled together in myth, but not in genetics. Consider the scale of what's actually being overturned here. This isn't a fringe claim resting on one unusual grave or one outlier skeleton. It's a structural finding about an entire founding-era town, backed by genome-wide data and isotope analysis across dozens of individuals. One town's diversity is a data point. A 2,000-year transect across the whole country is something else. In 2023, a study landed in Cell that dwarfed everything that came before it in scope. Rodriguez Varela and colleagues published The Genetic History of Scandinavia from the Roman Iron Age to the Present, in Cell, Volume 186, Issue 1, and the scale of the project set a new bar for the field. The work was led by a partnership between Stockholm University and Decode Genetics in Reykjavik, pairing archaeogenetics expertise with one of the largest population genomics operations anywhere in the world. That combination mattered, because the ambition here wasn't just to sequence more skeletons. It was to build a genetic timeline stretching across 2 millennia. The numbers back up that ambition. The team combined 48 newly sequenced ancient genomes with 249 previously published ones, bringing the total to 297 ancient genomes, spanning from the 1st century Roman Iron Age all the way through the 19th century. That's not a study of one era or one crisis point. It's a continuous record. They compared that ancient dataset against genotype data from 16,638 living people, drawn from Denmark and from every single county of Norway and Sweden. Modern genomes set against ancient ones, region by region, century by century. The authors themselves describe this as a full 2,000-year genetic transect, not a snapshot frozen at one moment in history, but a continuous record of how Scandinavia's gene pool actually changed, generation by generation, across war, trade, migration, and settlement. The dataset leans on sites already covered elsewhere in this story, Sigtuna and Birka among them, but it also reaches further back in time, into new material that predates the Viking-age entirely, including the pre-Viking Sandby-Borg massacre. To get pre-Viking DNA at all, the team had to go looking for the dead who broke Scandinavia's oldest funeral rule. Sandby-Borg sits on the Swedish island of Öland, a ring fort dating to the migration period, roughly 400 to 550 CE, centuries before the Viking Age even begins. It's a place defined by violence, not by raiding or exploration. Archaeological evidence points to an organized massacre carried out in the late 5th century. What makes the site unusual isn't just the killing itself, but what happened afterward. The bodies were left exactly where they fell, undisturbed, unburied in the customary way. That detail is the key to why Sandbyborg matters so much genetically. Iron Age Scandinavians typically cremated their dead, a practice that destroys DNA entirely, erasing any chance of genetic recovery. Sandbyborg's uncremated victims are the exception, offering a rare, direct genetic snapshot of pre-Viking southern Sweden that simply doesn't exist at most sites from this period. Around 30 individuals have been recovered so far, pulled from roughly 10% of the excavated interior of the ring fort. The dead span every age group, from infants to the elderly, a cross-section of an entire community caught in a single moment. One individual's remains carried something beyond human DNA. Detectable bacterial DNA from diphtheria. A disease signature preserved right alongside the human genome. A secondary record of illness surviving in the same bones. Genetically, the Sandbyborg victims trace back to local and regional southern Scandinavian ancestry. This is the before picture, the genetic baseline against which all of the later Viking-era diversity gets compared and measured. That baseline is what makes the next find so strange, because someone with foreign ancestry was already living in Denmark before the Vikings even started sailing. Among the 2023 study's 297 ancient genomes, one individual stands out for arriving far too early to fit the expected pattern. Her designation is VK213, a young woman excavated at Gerdrup, Denmark, radiocarbon dated to between 391 and 527 CE, placing her squarely within the pre-Viking migration period, the same window covered by Sandbyborg. Her genome carries the third highest British-Irish ancestry signal of all 297 ancient individuals in the entire dataset. A remarkable result for someone buried centuries before the Viking Age is traditionally said to begin. The paper's authors explicitly single her out, flagging her as one interesting exception, because the study's overall finding is that British-Irish ancestry doesn't spread widely into Scandinavia until the Viking period itself, centuries after her lifetime. Her presence changes the shape of the story. It means individual, isolated cross-North Sea contact was already happening in the 5th century, long before ships full of raiders started crossing regularly. The mass Viking-era migration wasn't the start of the connection between Scandinavia and the British Isles. It was simply the moment that connection became common, routine, and eventually overwhelming in scale. This complicates the tidy narrative that even some ancient DNA papers lean on for a clean story. The idea that the Viking Age itself opened the door to the British Isles. VK213 suggests the door had already been open, if only slightly, for generations before the first recorded raid. One woman, 3 centuries early. The pattern she's an exception to is the one that defines the entire Viking Age that follows. The 2023 study identifies 3 separate external sources feeding into the Scandinavian gene pool over its 2,000-year transect: the Eastern Baltic, the British-Irish Isles, and Southern Europe. Each of these 3 sources carries its own timing and its own regional footprint. They didn't spread evenly across Scandinavia, arriving all at once and settling uniformly into the population. Instead, each left a distinct geographic signature. British-Irish ancestry became widespread from the Viking period onward, appearing broadly across Scandinavia, rather than concentrating in any single pocket or region. It's the most diffuse of the 3 signals, touching Denmark, Norway, and Sweden alike. Eastern Baltic ancestry tells a very different story. It's far more localized, concentrated specifically in Gotland and central Sweden. This wasn't a Scandinavia-wide phenomenon. Sweden's own geography absorbed this particular influx, not Denmark's coastline and not Norway's fjords. This finding lines up precisely with the 3 genetic diversity hotspots the Margarian 2020 team had already flagged in their own research: Denmark, Gotland, and Öland. 2 of those 3 sit within modern Sweden, reinforcing a pattern now confirmed across separate studies using separate methods. The pattern gives a geographic answer to a question the sagas themselves never could resolve. Which parts of Scandinavia were actually the most cosmopolitan? And the answer isn't the mainland capital regions most people assume, the centers of political power. It's the islands and the border zones, the places built for contact rather than command. Gotland's Baltic connections weren't just cultural. They were, quite literally, written into the island's DNA, and they went both directions, deep into what's now Russia. Staraya Ladoga sits in Russia, 128 kilometers east of St. Petersburg, and it stands as one of the earliest and most important Viking-age trading hubs anywhere to the east of Scandinavia. The route connecting it back to Scandinavia isn't a modern archaeological reconstruction. It's named in a real historical source. Nestor's primary chronicle calls it the way from the Varangians to the Greeks, a route running from Ladoga along the Volkhov River, through Lake Ilmen, down the Dnieper, out to the Black Sea, and finally to Constantinople itself. Strontium isotope analysis of 15 burials from Ladoga's medieval St. Clement Church Cemetery, dated to the 11th and 12th centuries, identified 5 non-local individuals. Their isotope signatures specifically match the Mälaren region of east-central Sweden, not Scandinavia in general, but one particular Swedish valley. Genetic sampling from the wider Viking genome dataset adds a named individual to this picture, VK23, recovered from Ladoga and dated to the 10th through 12th centuries, carrying Y-DNA haplogroup IM253 and mitochondrial haplogroup U4A1A. The historical record and the genetic record meet here. The founding of the Rurik dynasty is traditionally dated to 862 CE, with Norse settlers credited by the primary chronicle as central figures in the earliest Rus state. The genetic and isotopic data don't just support that old story of Viking founders in Russia. They go further, tying it back specifically to one Swedish region, the Mälaren Valley, rather than to Scandinavia as an undifferentiated whole. Sweden's DNA didn't just travel west into England and Ireland. It traveled east, all the way to Constantinople's front door. Zoom back into Sweden itself, and the picture only gets more detailed. Named Viking-age genomes recovered from Varnhem near Skara add more direct data points to a map that was already filling in. VK29, recovered from Varnhem and Skara and dated to the 10th through 12th centuries, carries Y-DNA haplogroup IS7642 and mitochondrial haplogroup T2b3b. A second individual, VK30, from the same site and the same time frame, carries Y-DNA haplogroup RS2857 and mitochondrial haplogroup U5b1c2b. 2 more named people, 2 more genetic fingerprints, added to the record. These individuals don't stand alone. They sit alongside the samples from Sigtuna, Birka, and Sandbyborg as part of the broader ancient Scandinavian reference panel used across both the 2020 and the 2023 studies, part of a growing body of evidence rather than an isolated curiosity. The regional spread across all these sites, Öland, Gotland, Uppland, Sigtuna, and now Skara, is what actually let researchers detect Sweden's internal genetic structure. Without that spread, without genomes drawn from so many different corners of the country, scientists would have had no choice but to treat Sweden as one uniform population, a single genetic block with no internal variation worth measuring. That internal structure turns out to matter more than most people assume. It isn't a footnote or a technical curiosity buried in a supplementary table. It's the foundation for the study's most quietly explosive modern-day finding, the discovery that gives this whole genetic transect its sharpest edge, because that same internal structure hasn't disappeared. It's the reason 2 people from opposite ends of Sweden today can carry a strikingly different genetic signature. The 2023 cell study identifies a north-south genetic cline running through modern Scandinavia, a gradual, measurable shift in ancestry as you move from southern Sweden and Norway up toward the northern reaches of both countries. The main driver behind that cline is differing levels of Uralic ancestry, a genetic signal linked to populations from further east and north, historically associated with Finno-Ugric-speaking groups. This isn't a subtle statistical artifact. It's a measurable gradient, visible in the genomes themselves. Crucially, the study finds this cline was already present during the Viking-age, and possibly earlier still. It isn't a modern artifact produced by recent migration or 20th century population movement. The north-south divide in Scandinavian ancestry has deep roots. This connects directly back to the earlier Sigtuna finding. The I2A and N1A haplogroups, absent from modern Sweden and Norway, but recovered from Viking-era Sigtuna, were themselves linked to Finno-Ugric ancestry, the same genetic thread running through this north-south cline centuries later. The modern numbers put a figure on the pattern. Norwegians today carry roughly 12 to 25% North Atlantic ancestry. Swedes carry roughly 10%. Real, measurable differences, distributed unevenly across the population. Put together, this means Sweden's modern regional identity, the lived sense that northern and southern Swedes are somehow different from one another, has a real, 1,000-year-old genetic backbone behind it. Just not the backbone 19th century nationalists imagined when they built their myth of a single, unbroken bloodline. So the ancestry variation is real. It's just running in a completely different direction than the myth ever claimed. Here's the finding the 2023 team highlighted as genuinely unresolved. A puzzle they put on the record themselves rather than one uncovered by outside critics. In some regions, modern Scandinavians carry lower levels of non-local ancestry than the ancient Viking and medieval-era genomes recovered from those same regions. Ricardo Rodriguez Varela stated it directly. Although still evident in modern Scandinavians, levels of non-local ancestry in some regions are lower than those observed in ancient individuals from the Viking to medieval periods. In plain terms, the foreign ancestry surge of the Viking Age didn't simply plateau once it arrived. In places, it partially receded over the centuries that followed, leaving modern populations with less external ancestry than their medieval ancestors actually carried. The study's authors are explicit that the reasons behind this aren't yet clear. This is presented as an open question in the research itself, not a solved mystery with a tidy explanation attached. One possible read on the data. Ancient immigrant populations may have contributed proportionately less to the deep modern gene pool than their visible Viking-era presence would suggest, meaning some lineages simply didn't persist across 1,000 years of subsequent generations, births, and demographic change. The implication flips the popular story on its head. Sweden wasn't at its most genetically pure in the deep past, only to be diluted gradually over time by later contact with the wider world. Genetically, in some measures, the pattern runs the other way entirely. That's the finding scientists keep having to explain very, very carefully, because of who's been listening. The pure Viking bloodline idea isn't just an outdated historical assumption, quietly corrected and set aside by modern scholarship. National Geographic's own 2020 coverage of the Margarian study states plainly that the myth is celebrated today among various white supremacist groups, used as a justification for claims of racial superiority. Geneticists have addressed this directly and publicly. A 2022 comment piece in Nature, titled "Counter the Weaponization of Genetics Research by Extremists," describes population genetics papers being repeatedly cited by neo-Nazi and white nationalist online communities, misread and redeployed in service of an ideology the research itself contradicts. A UCLA sociology study conducted by Panofsky and Donovan tracked how white nationalists actually respond when their own consumer DNA test results show mixed or non-European ancestry. The pattern was consistent, mostly by disputing the test's statistics or by claiming some kind of conspiracy, rarely by updating their views in light of the evidence in front of them. This is the real context behind why Swedish researchers like Götherström state their findings so bluntly and so publicly. The Swede doesn't exist, genetically, isn't just a punchy quote built for a headline. It's a direct rebuttal aimed at a specific, documented misuse of earlier genetic research, spoken by someone who understands exactly how that research has been twisted before. None of this changes the underlying data. The diversity documented at Sigtuna, Birka, Salme, and across the full 2023 cell dataset is measured, peer-reviewed, and independently replicated across two major studies conducted 6 years apart. What it changes is the stakes involved in publishing it. This is genetics research that has had to actively defend itself in public, on the record, against people trying to read it backwards. Which is exactly why the open questions the researchers themselves admit to matter, because half-finished data is exactly what gets exploited. Step back and the shape of the evidence becomes clear without needing to be recited line by line. Two major studies, 6 years apart, hundreds of ancient genomes, tens of thousands of modern ones, all converging on the same structural picture: mobility, mixture, and regional variation, not the uniformity the old myth demanded. Something genuinely remains unresolved, and the researchers themselves say so in their own words. Why non-local ancestry receded in some regions after the Viking Age, instead of simply persisting the way a straightforward story of migration and settlement would predict. The stated next direction for the field points forward rather than backward. Finer-grained regional sampling, especially in currently undersampled areas, combined with isotopic mobility data, and eventually, multi-generational tracing of how Norse settled communities actually changed in the centuries after arrival. The Sigtuna quote can stand here one final time as the episode's anchor, without repeating it verbatim as a crutch. It's the sentence a Stockholm professor was willing to say to a national newswire, on the record, knowing exactly how it would land. If the DNA of the people who built Sweden was never one bloodline to begin with, what exactly is left of the myth that said it was? The genome from grave BJ 581 has been out of the ground for over 140 years. Science only started listening to what it actually said in 2017.